Hello, bonjour, edate. My name is Carolyn, and I'm a fourth-year PhD candidate at Queen's University in Kingston, Ontario. My research is broadly interested in validating remotely sensed vegetation productivity trends by combining field investigation with dendrochronology. Dendrochronology dates past events using the annual growth rings of woody plants. Try it out. How many rings can you discern in this Arctic shrub? How old is this shrub? Once you've counted, you can share your answers in the discussion to follow. The research I'm about to share will demonstrate a few things we can learn from counting rings, which is one of the simplest applications of dendrochronology. My talk today will review recent findings on shrub growth patterns and characteristics for a study area in the central mainland Canadian Arctic, where I'm trying to tease out whether tall shrubs are the main contributor to remotely sensed greening trends. My research interests are driven by four fundamental changes that have been taking place across the Arctic in recent decades. First, the Arctic climate is warming 3 times faster than the global average. And concurrent with this rapid climate warming has been an increase in vegetation productivity, also called greening, across the tundra, as detected through remote sensing. And third, while caribou populations are known to fluctuate in decadal cycles, most barren ground caribou populations are now in steep synchronous decline. Another substantial change taking place across the Arctic, the timing of which coincides with those of warming, greening, and declining caribou numbers, is rapid shrub expansion. This phenomenon manifests itself in several ways. Existing shrubs are becoming larger and taller. Shrubs are colonizing new areas, both within and beyond their range, and/or areas with existing shrubs are filling in and becoming more dense. Shrub expansion has been shown to alter surface albedo, moderate the effects of wind, snow, and erosion, change microclimate and nutrient cycles, compete with other tundra plants, and affect interactions with herbivores, pollinators, and soil microbes. So understanding shrub expansion across the tundra is therefore not just important, but necessary for understanding the consequences of climate change in the Arctic. The Bathurst caribou herd of central Canada has declined 99% since its last population peak in the 1980s, and a recent remote sensing study found that the range has experienced greening. While much is known about greening in coastal areas, there's a limited understanding of the causes and consequences of greening at inland sites. Tall shrubs are those that grow erect rather than prostrate. And given that tall shrubs are the dominant plants on the Bathurst Range, and they've been identified as strong contributors to tundra greening in other regions, I hypothesized that greening on the Bathurst Range is the result of a change in the structure, composition, and/or growth of tall shrubs. This study was conducted just beyond treeline, an important late summer feeding area for caribou, but also where some of the strongest greening trends within the Bathurst Range have been detected. Furthermore, the forest tundra transition zone is experiencing greening more broadly across the global Arctic as well. From available mapping of remotely sensed greening trends, I chose sites that have experienced significant increases in vegetation productivity for comparison with sites where productivity has not changed over the last 2 decades. The map in the bottom left shows our 2018 expedition route and sampling locations within areas of no change, those are in orange, and what we call greening, in green. Shrubs were sampled from 44 replicate points within each of 15 greening and 14 no-change sites. In my study area, the tall shrubs are dwarf birch, Betula glandulosa, and various species of willow, Salix. At each site, I noted whether any tall shrubs overlapped each sampling point so as to estimate percent cover. I systematically measured distance from a point to the nearest living stem, noting the species. From this, I can estimate density and relative abundance of birch to willow. I clipped that stem at its base and collected a sample. By counting growth rings in the lab, I can find out when that stem became established and also estimate its minimum age. I measured the length of that stem and collected a second sample halfway up the stem. By counting rings at midpoint, I can obtain estimates of the stem's growth rate. I counted the number of living and dead stems associated with each sampled individual. And back in the lab, student volunteers measured the diameter of all of the stem samples, which is useful for exploring woody growth. This simple sampling method, which combines field-based measurements with lab-based growth ring counts, allowed me to examine several metrics related to stem establishment, distribution, and growth, and to compare these between greening and no-change sites. A few points to note. First, in our study area, willows tend to grow as distinct individuals emanating from a central root collar, whereas birch, by contrast, tend to form multi-stem mats of clones. I'll use the term genet to describe both an individual or a group of clones believed to have originated from a single seed. Now, second, we can't detect stems that established, died, and were no longer present at the time of sampling. And thus, the dead stems I counted and will be speaking about are the persisting dead stems. And third, by clipping stems at ground level, the number of rings translates into that stem's minimum age, as there could be growth just below the ground surface, and shrubs don't necessarily put on diameter growth every year. But for simplicity, I'll simply say age. And so here is what I found. There was no difference in mean stem age, age of the youngest stems, or age of the oldest stems between greening and no change sites. And together, these metrics target the central area and both tails of the distributions of establishment to suggest overall similarity in stem establishment patterns between site types. Both greening and no change sites supported an average density of 37 stems per square meter, which can be visualized as about one stem, maybe about a hand's length apart within a grid. Tall shrub cover was found to be nearly 13% greater at greening sites. And while birch outnumber willows in my study area by about 9-to-1, the proportion of birch to willow did not differ between the two site types. And stem length was also similar across both site types, averaging about 48.5 centimeters, thigh height or so, if pulled up straight. There was no difference in stem growth rate between greening and no change sites. On average, stems grew at a rate of about 3.3 centimeters per year along their full length. There was also no difference in the relationship between stem age and thickness at its base. For every year of growth, the diameter of a stem's base increases by about 0.1 of a millimeter, the thickness of a human hair. The number of living stems did not differ between greening and no change sites. However, genets at no change sites consistently had 9% more persisting dead stems compared to those at greening sites, regardless of overall size of the shrub. And by size, I'm referring to the number of stems, live stems associated with that shrub or genet. So taken together, in light of trying to tease out the contribution of tall shrubs to remotely sense greening trends, characteristics of shrub patterns and growth are very similar across the two site types. More cover at greening sites suggests greater photosynthetic output. But where is this coming from? There's no difference in the length or number of living leaf-supporting stems. On the dry, cold tundra, where dead woody material takes a long time to break down, we found evidence that stem mortality is lower at greening sites, and less mortality could indicate healthier, more productive shrubs. Considering that remote sensing relies on bands of reflected light, even a slight increase in tall shrub cover can dominate the greening signal. However, reflectance from vegetation in areas with more non-photosynthesizing surfaces, such as dead stems, will blend with adjacent bands of light to moderate the greening signal. So are tall shrubs the main contributor to remotely sensed greening trends on the Bathurst Range? Well, despite no difference between greening and no change for many of the metrics we investigated, it does appear that yes, tall shrubs are a contributor. In terms of light reflectance, more cover at greening sites is reflecting more green light, while dead stems at no change sites are dimming the green reflectance at those sites. And together, that results in the productivity differences detected by satellites. However, future research should consider leaf area and number of leaves or above ground biomass, as well as stem branchiness and other factors that would result in a greater photosynthetic ability of tall shrubs. Our data explorations also confirm that the distribution of shrubs is patchy, something that could confound our assessments of density and cover, so this too should be explored further. Regardless, my study offers a snapshot into the landscape traveled by the Bathurst caribou. I've set the stage for upcoming investigations into the contribution of other plant functional types to greening, particularly preferred forage such as lichen and graminoid, and for analyses of caribou movement and habitat selection relative to increased shrub cover. Together, these insights will help us better understand how changes in plant communities may be related to the current population declines of barren ground caribou. This research was made possible by many hardworking field hands and with funding provided by many partners. And with that, I would like to thank you, my audience, for your interest and attention today.